Hello, everyone. I'm Eric Camino. I'm PPMD's Director of Research and Clinical Innovation. Today, I'm joined by Rachel Schrader, who is our SVP of Clinical Care, and Dr. Tim Kripe from Nationwide Children's Hospital. Dr. Kripe is Chief of the Division of Hematology and Oncology at Nationwide Children's Hospital, a Gordon Teeter Endowed Chair in Pediatric Cancer at Nationwide Children's Hospital, and a Professor of Pediatrics at The Ohio State University. With the recent tragic news of the loss of two patients in the myotubular myopathy gene therapy trials, PPMD has seen a number of questions from the community concerning AAV and dosing in our current gene therapy clinical trials. And we felt it would be beneficial to the community to provide some foundational information on those topics, which are often discussed but may not be thoroughly explained. So Dr. Kripe has graciously agreed to help us answer some of those questions that we've collected from the community. So Rachel and I will be posing some of those questions and providing some more information. And so I think, well, first of all, Dr. Kripe, thank you for joining us today.
Happy to be here. Thanks for having me.
And I think a great place to start is really with AAV. When these clinical trials are discussed, we often hear about different types of AAV. We hear, AAV9, AAV8, AAV RH74. And they're always talked about in terms of the different serotypes and the tropism they have, but we don't always dive into what that actually means. And so we wanted to make sure that the community has an understanding of what that terminology is. And so if you could give us a bit of a breakdown of what is meant when we say a serotype of AAV and what we mean by tropism.
Yeah, sure. I kind of thought you might ask that question. So I actually have prepared a few slides for you to talk about that issue. It's a very important, critical issue to understand. So in terms of serotypes and tropism, what is a serotype? So if you look at the definition, a serotype, as defined by dictionary.com, is a group of organisms, microorganisms, or cells distinguished by their shared specific antigens, as determined by serologic testing. So what are antigens in serologic testing? Antigens are what are recognized by the immune system, usually by antibodies or T cells. And in this case, mostly by antibodies. And serologic testing means if you get infected with one type of virus or one type of bacteria and you get an immune response to that, a different serotype is usually one of that same class of virus or bacteria, but that's not recognized by the antibodies of the same type. So in other words, different serotypes usually generate different kinds of antibodies and can be distinguished in that way. And so it gets a little more subtle and nuanced with AAV though, and other viruses, because there are many subtle differences that may be considered different serotypes where the immune reaction actually does cross-react and is shared against those viruses. So let me just show you a little bit more detail of what I mean by that. So for example, AAV is shown here, represented, the line here and the boxes represent the genome of AAV. It's about 4.7 kilobases or 4,700 base pairs shown by that line. And there's two major genes encoded by that genome, the rep and cap genes. And the cap genes determine the capsid structure of the virus, and that's what's exposed to the immune system. And so if you look just, for example, at a small area of this gene from different serotypes, serotype 2, 5, or 6, and you look at the DNA sequence, and that's the A, C, G, and T letters, and those, every 3 sequences encodes an amino acid. And the amino acid is what makes up the protein. You can see that these 3 different serotypes differ in their gene sequence, and in some cases that doesn't make any difference in the amino acid. All these same 3 encode proline amino acid. But in some cases, it makes a difference. So for example, this change here, you get an arginine instead of a lysine here, the R instead of the K. So these differences in amino acid sequence in the capsid shell are what arise or what give rise to different serotypes. And this is shown here by some really nice structural biology work where the amino acid differences can result in real structural differences. Now, this is computer-generated representation of the structure of the outside of 3 different serotypes, 2, 4, and 8. And you can see that they're very similar, and it's sort of like those pictures where you're trying to look and see what's different, in this image of a person, and what's the difference between that and this image, and you discover, oh, the person doesn't have their purse in this image. So what are the differences? They're very subtle, but you can see some of them. For example, if you look at this green structure here on AAV2, it looks a little bit different than the same general shaped green structure here. There's a little bit more blue in between some of those greens. So very subtle differences in structure, but those subtle differences can result in real biologic differences in how the virus behaves. And how the virus behaves can be among a lot of different things, but one of them is tropism. So what does tropism means? Well, the formal definition is an orientation of an organism to an external stimulus as light, especially by growth rather than movement. So a plant that's trying to grow toward the sun, that's the tropism. So where does it like to go? What areas does this organism like to be in? So tropism in virology is really what cells does it like to infect, or can it infect? And when it comes to the body, what cells does it actually go to and get into? So that's what we mean by tropism. Is it going to the liver? Is it going to the muscle? Is it going to the lung? And how much does it go to each of those spots? And the relative amounts that it goes to each of those spots is called biodistribution. So different serotypes can exhibit different tropisms. This is an example from a while ago in some mice where they gave an AAV that expressed light, essentially expressed light, and they could detect where it went and detect it at different times. So this is an image with different serotypes, 1, 6, 8, 7, and 9, and over time, 14 days, 29 days, etc. And you can see where the virus went is very different from serotype to serotype. It's similar for all three with the same serotype. This looks like AAV is mainly going to the liver, but with AAV6, you're getting some other spots showing up in different parts of the body, and in different amounts. So at the end, you can see down here by day 100, you got a very different distribution for each serotype and different amounts. The red indicates really high amounts compared to the purple. And same over here. Although they're similar to each other within a category, within a serotype, they're very different from each other, both in where they go and the amounts that they go there. And that's a result of the subtle differences in amino acid sequences mainly of the capsid proteins that make up that capsid shell of the virus. So this can be very deterministic in terms of the choice of serotype and what it's doing. So interestingly enough, there's really an unlimited number of possible serotypes. There's a lot that exists naturally in nature. The ones that have come out of human cells are usually just called AAV with a number. The ones that come out of rhesus monkeys are called AAV-RH with a number, like RH74. And there's a lot now that are being made synthetically because we have the ability with recombinant DNA technology to change the amino acids. We can change them randomly. We can change them at will. We can select for those that go to certain organs, and so there's really an unlimited number of possible serotypes. So lots naturally occurring, but we can also engineer those. And that can determine then the differences where they land in the body, the biodistribution, the tropism, how well they get there, how much they get there. Also, what degree of immune response there is, and whether that immune response inactivates them or not, and whether that different serotypes are inactivated by the same antibodies or not. And then it can also determine the amount that's produced during manufacturing. So it can have an effect on the costs and the manufacturing. So that's just an overview of the differences of serotypes and tropism. I hope that answered your question.
I think that was a great explanation that helped provide a lot of context.
So, Dr. Kripe, I've got a question for you. I think that graphic with the purple and the red and the different body parts is really helpful for me to understand how the different serotypes are different. But could you help me understand a little bit about how a particular serotype or virus is selected for any specific trial?
Yeah, that's a great question. And I think a lot of it starts with what the goal is of that trial. So if the goal is to get into muscle cell, you want one of these that does reach muscle. Not all of them get into muscle cell. But with that said, we have to understand that none of these are really that specific. they're going to go to multiple places. Now, people are trying to make them as specific as possible. And as we continue to get better at engineering these or even selecting them, then we may be able to reach a point where you get 90% going to a specific organ. But it's important to remember there's a lot of different determinants of that. It's the receptors or the locks on the cells that the virus finds that determine that. And so you have to find the right tweak on the virus so that it can only go to that receptor. But sometimes those receptors are expressed in other tissues, so it may be impossible to make it really specific for a given tissue. And so with the naturally occurring ones, people have said, well, let's figure out where each goes and then pick the one that goes where we need it to go the best. But almost all viruses, because they're particulate, they're particles, they actually get soaked into the body's filters, which is like the liver and the spleen. So they're going to end up there no matter what, to some degree, just because of the physical nature of viruses as opposed to small molecule drugs. So that's one issue we have to contend with. So even if we think, oh, this one goes to the liver, it's not just going to the liver, it's also going to go elsewhere. But if we want one that just goes to the muscle, it's also going to go to the liver, for example, and maybe other places. So we have to keep that in mind. But we can select these to be more favorable toward what our goals are. If we want one to go to the brain or nerve cells, we can find those that are preferential for that, and try to engineer some that even do it better. So that's the first, I think, level of selection of why you might pick one, to make sure it's going to end up where you need it to end up. There are other reasons to pick different ones. And those may be, if there are some that are not just targeting where you want them to go, but detargeted from where you don't want them to go to try to minimize side effects, for example. But I think the biggest rationale for selection is tropism. Another big rationale for selection might be the natural prevalence of antibodies against these. These are viruses that are not associated with disease. It's important to differentiate adeno-associated virus from other viruses like adenovirus. Completely different viruses, even though they have a common name. Adenoviruses do cause disease. They cause colds, conjunctivitis, that sort of thing. But adeno-associated viruses are a completely different class of viruses, and they don't cause those diseases. Nevertheless, those viruses are percolating throughout the human population. And so a certain percentage of people have pre-existing antibodies. They've been exposed to these viruses. They don't know it. They don't cause disease, but they've been exposed somewhere along the line, and they've generated antibodies against them. So you may want to not pick a virus where there's very common to have antibodies against. I think that's one attraction sometimes for some of the ones that have come out of monkeys, because there's fewer people that have antibodies against those viruses pre-existing. But certainly not everyone has antibodies to any particular one. And there might be ways around that we can talk about, but that might be another criteria for selection.
Oh, great.
That's incredibly helpful. Thank you.
And actually, just to touch on a point that you just expressed, with the tropism of these, the different viruses, although there's a tissue that maybe they have a higher prevalence of targeting, as you did express that it can go to other tissues in the body. Could you maybe explain a little bit about then what the role of the promoter is to ensure that we're not expressing the transgene in targets where we don't want it to be?
Yeah, sure. So what's shown here is a wild type, what we call normal, naturally occurring virus, but the recombinant versions that are made for gene therapy purposes actually are what we call gutted viruses. That means this gene, the replication and cap gene, have been removed. They're actually moved onto a separate plasmid or other construction such that those proteins are provided into the system when you're making the virus, but the genome that gets encapsulated in that capsid shell that's actually in the virus producing these gene therapy products does not look like this. It has these ends on it, but the inside here is whatever we put in it. So a dystrophin gene or other gene, hemophilia gene. So these have been replaced. And why was I going this way? What was your question again?
With the fact that the different viruses have
Promoter.
different, right, tropism, how do we ensure with the promoter?
Yeah, so what goes in here, sorry about that lapse. What goes in here then is the gene of interest, but also it's the on switch for the gene of interest. So the promoter, what's not shown here is each of these has its own promoter, and that promoter basically instructs the cell to start making the gene right there and to turn it on, to start making the protein, to transcribe the gene, the RNA from the DNA. And that promoter is something that then is inserted into the gene of interest that's put in here, like the dystrophin gene. And for example, the dystrophin gene, most promoters, then they're using a muscle-specific promoter. So promoters are basically the on switch, and the on switch is can be one that's either ubiquitous, that it's on in any kind of cell that you put it in, or it can be what we call a tissue-specific on switch, or a tissue-specific promoter. And those promoters tend to be active only in certain cells. Now, it is very unusual to get a promoter that's active only in a very specific type of cell, but that's completely on in one cell and completely off in all other cells. But we can get pretty close to that. So these muscle-specific promoters are largely turned on in muscle cells and turned off in non-muscle cells. And that's really what makes these viruses muscle-specific. The virus itself isn't just going to muscle, but it's turning on that gene selectively in the muscle and making the expression of that protein relatively muscle specific. So the virus is going a lot of other places, and it could cause side effects in those other places just from being there, but it's only turning on the dystrophin gene in the muscle cells. And certain promoters might only do it on in skeletal muscle versus heart muscle, and some will turn it on in all those muscles or smooth muscles. So the selection of the promoter is also important in the success of these viruses.
Okay, so now that I understand a little bit about different serotypes and where they go and how they can be somewhat tissue specific depending on the promoter, maybe you could help me understand a little bit about the dose and how much virus we're delivering in these gene therapy trials. We hear about high dose and low dose, and you see something like E integrated into that dose. What does that mean?
Yeah, so again, I'm one step ahead of you. Got some slides ready. So the E in the dosing means exponential. So unfortunately, it takes us all back to our math days. What may have used to have been high school, some middle school, and probably now elementary school. So the E is exponential. We think of the virus dose in terms of folds of 10-fold or logs base 10. And these numbers can be very staggering. So, and the reason they're staggering is because the goal here is to try to get the virus into every cell in your body. So if you've got billions or trillions of cells, you've got to have billions or trillions of viruses. And in fact, you need more than that, because getting a virus into the cell isn't the most efficient process. So you usually need more than one per cell. And to get it to have the chance to make it work. There's a lot of places along the line of a virus kind of entering a cell and getting into the nucleus and expressing the gene that could go wrong. And so you got to hedge your bets and have enough of them around so that at least one makes it. And so the numbers are quite staggering when you're trying to reach that many cells in the body. And so here's an example of some of the actual numbers. So intravenous dosing for AAV, the FDA-approved virus for spinal muscular atrophy is given at 1.1 e to the 14th VG per kg. So vector genomes per kilogram. So this is per body weight. So if you weigh more, you get a higher dose. The dose is the same in terms of per kilogram, but higher total dose of vector genomes. Vector is just the virus. Genomes is that DNA sequence I showed you before, and we can count the number of those that's in a given solution of the virus in a given prep. And as an aside, each prep has some empty particles. So those capsids I showed you, it's not 100% efficient in terms of packaging them when we produce these viruses. And there's debate about those in some ways may be good because they can absorb the antibodies and be kind of a decoy, and in some ways they may be bad because they can be causing responses and other side effects. So that's just an important side to keep in mind. But of those that actually have vector genomes, there's 1.1 times 10 to the 14. And we'll talk more about what that number means in a second. Some of the trials going on with muscular dystrophy are also using the same order of magnitude, e to the 14. So that's 10 to the 14. So that's a 1 followed by 14 zeros. And we'll talk more about that in a minute. And that's in contrast to some of the studies with hemophilia where they're only using 6 to the 13 or 6e to the 11th. So 6e to the 13th is half the dose of Zolgensma, and 6e to the 11th is 0.5% the dose of Zolgensma. So that could be advantageous at a lower dose, potentially causing fewer side effects, less cost. You can get more patients dosed with a given manufacturing run. So that can be important if we can use a lower dose. But the reason we can get away with lower doses in hemophilia is they're not trying to hit every cell. They're not trying to hit or get the gene into every liver cell. You only need it in enough percentage of the cells to secrete the product into the bloodstream that they need to fix hemophilia. Whereas in SMA or Duchenne, we need a virus in most, if not all, of the muscle or nerve cells. And so you need a higher dose to ensure that happens. But these numbers are just a number on the page right now. But what do they really mean? I mean, these are actually very staggering numbers. So let's put that into context. So vector genomes, also sometimes called genome copies. 1 e to the 14th or 1 times 10 to the 14th per kilogram of body weight. So just, again, your math with logarithms, 2 e to the 14th is twice as much as 1 e to the 14th. 5 e to the 13th is half as much because we're now the log below, tenfold below, but now we're doing 5 of it. So if you go 5 times 2 is 10, that's the same as 1 times e to the 14th. So 1 followed by 14 zeros, that's 100 trillion. So I calculated out, that's a round trip in miles to the sun for 1 million trips. Or it's the number of seconds in 3 million years. And it's only 5 times the national debt, so that tells you something about our national debt at 20 million right now. So these are staggering numbers, very staggering numbers. And I think the reason it's important to understand this is because we wonder, well, why are there side effects? We're putting a lot of foreign protein, even if it weren't a virus. We're just putting a lot of foreign protein into the body when you do this. And we can get away with it because these are very small particles, and these are small. So the volume of what we're putting in is not that great, but it's staggering numbers of particles. But it's not surprising that they might cause some side effects. So just to save you the question, it does have side effects, and I think it's mainly because we're having to put in these large numbers. And this is just from a recent online article commentary. The link is at the bottom, but it shows you some of the different conditions that have been treated with gene therapy and the doses. Most of it in the high 10 to the 13th or e to the 13th, some e to the 14th. This shows you the serotype capsid that was being used, and then some of the adverse events. And we can talk about these in detail and why we might be getting some of these different adverse events or side effects. But it's just my point here for this question is it's not surprising that putting that many particles in someone's body is actually causing some problems, and that there's a reaction on the part of the body.
That makes so much sense.
Side effects if you want. But.
No, actually, I think that that might, with the serious adverse events that you just listed, that might be a great spot to kind of dive into. Obviously, some of the questions that we've received from families is concern due to those 2 serious adverse events that we had in the myotubular myopathy trial that, as I mentioned at the start of this conversation, did result in the death of those 2 children, which is tragic. So I think if maybe we discussed a little bit about what these adverse events are that we may be expecting, and what some of the strategies that we have to help maybe mitigate these adverse events that we may be seeing.
Yeah, so let's talk about them a little bit. So, there's the SMA trial. N of one means one patient had this in that particular trial, and N of one in this particular trial. Elevated liver enzymes, as we just described, all of these viruses go to the liver. It's the major filter of the body. You put something in the bloodstream, it's got to pass through the liver, and it might get taken up there and cause damage, may actually kill some of the liver cells. Now the liver regenerates, and that's a plus. And so sometimes this can just be a transient effect, and can heal itself. So that's really not unexpected. And there are things we can do to try to minimize that or deal with that. And that's not untrue for lots of drugs. Lots of drugs, the liver is the filter, the liver is the metabolism center for eliminating drugs from the body. And so to have effects on the liver is not uncommon. We deal with it all the time in medicine. I guess it's when it gets severe that can be a problem. So even Tylenol is damaging to the liver. The myotubular myopathy study, as you mentioned, has had some side effects like any drug might, some GI problems, elevated troponin in this one patient, so that suggests there might have been issues with the heart. But more seriously was the liver inflammation, hyperbilirubinemia in 3 patients, and potentially resulting in bacterial infection and sepsis that resulted in the death of 2 patients. So that is obviously the biggest concern we have now. What might be causing that? And we don't have a lot of information. A lot of it's not out in the public domain yet. But this is a disease we know where patients can have underlying liver problems to start with. And we understand that these 2 patients did, in fact, have underlying liver problems as part of their normal disease. We don't know anything about the details of those and how severe and what extent. And so one question would be, if those patients happen to be more severe than others, maybe we can have a cutoff of eligibility, if that puts you at too high of a risk. So there's certainly going to need to be much more understanding about what happened in those situations in terms of their reaction to the virus. And also, frankly, why it caused the bacterial infection and sepsis. Was it the liver damage that led to that, or was that the bacterial infection and sepsis actually a separate, distinguishable event from the liver problem? And I think we just don't have enough information about that to know. What we can say, though, is, and we also don't know whether it's due to the different serotype. This is AAV8, but obviously there's other studies with AAV8, and some not listed here, that are the hemophilia studies have been using AAV8, where patients have been dosed safely. Whether it was due to the high dose, again, these you asked about low dose and high dose, and I guess I forgot to answer that in the prior question, but everything's relative. Those doses are staggering, I showed you. So they're all very high. There are some that are higher than others. So it really shouldn't be low dose and high. It should be high or super high or even higher kind of descriptors. Even the hemophilia doses at 10 to the 11th or 10 to the 13th, they're lots of protein and fairly high doses, but compared to these doses are much lower, tenfold, hundredfold, like I said, 0.5% of the Zolgensma dose. So those might be considered relatively lower doses. But those can also cause side effects. But certainly we think, just like any drug, the higher the dose, the more likely there is side effects. And that's no different with this. So these 2 patients were in the high dose, and so was that the 3e to the 14th. That's probably bordering on what the human body can tolerate, frankly, for a foreign substance to be put into it. So, I think we just need to better understand what happened in those cases and how we can mitigate against it. We know that usually we can mitigate against liver toxicity by giving steroids. Because much of the liver toxicity is due to an immune response to the virus itself. And one thing we didn't touch on is, once the virus has delivered its payload, delivered its gene, it gets cleared. the proteins that make up the virus capsid are still hanging around for a while, and you can get an immune response against those. But eventually they get cleared, and long term, there's no gene making those proteins around. The gene is making the dystrophin protein, but not the viral protein. So those virus proteins get clear, and they shouldn't be a problem long term for an immune response. But that interim period, they can induce an immune response. So we can mitigate against that by using drugs that suppress the immune system, such as steroids. And finally, the final point I'll make just before I let you ask more questions is, there are other side effects like reduced platelets and red blood cells and complement activation and acute injury. Again, you're putting these particles, these foreign proteins into the system. They're going to react to it. And complement is part of the immune system. And the complement system is designed to attack bacteria and viruses and to punch holes in cells that shouldn't be there and destroy them. And so they're activated in a lot of different ways, partly by antibodies. And so if you get antibodies against the virus and you can start to activate complement, you can have side effects that come from complement activation. And that also includes platelets being consumed, being clumped, being activated, and their numbers going down. So these are not unexpected from the use of these kinds of viruses, but they are things we need to deal with better.
Okay, so Dr. Craig, you mentioned as you were talking through this chart that there are high dose and low dose, or high dose and super high dose. And then you also mentioned that maybe there's like a maximum amount of particles that a human body could even handle. And I thought that that was really interesting. Could you help me understand a little bit more about how we come to that high dose and low dose that's, seen in any given trial, or that high dose and super high dose? Where do we get those numbers from?
Yeah, it's a great question. So normally in drug development, especially in my field in oncology, we try to start with super low doses, especially for first in human studies. And we dose escalate bit by bit, usually 3 patients at a time. And we're not usually going up tenfold like some of these studies. We're going up, 20% at a time or so. Slow escalations, but it also might mean there's a dozen different dose levels before we get up to levels that either are causing side effects or are even an effective dose. So the downside of that strategy is that most patients that are in the early cohorts of clinical trials, phase 1 cohorts where we're dose escalating, they're not getting levels or drug doses that are effective. And in many of those cases, they can either go on to have other drugs or circle back around to get that same drug later when it's approved or we know the better dose. In gene therapy, because of this immune response, and at the moment we don't have a great way to do any redosing. Once you get an immunity to that virus, it's with few exceptions, it's impossible to get that virus again. And therefore the consequence of starting with a low dose and exposing someone to the virus and then not allowing them to get it again, or putting them through this without the prospect of benefit because you're using a super low dose is really not very attractive or ethical. So the gene therapy field and the FDA, who regulates the gene therapy field, have adopted a strategy or an attitude that we want to try to come as close as possible to a good therapeutic dose from the get-go when we first start testing patients. So we need as much information as possible from laboratory studies and animal models to try to narrow in on what a not only safe dose is, but an effective dose to start with. So the dose we've arrived at is based on studies in animals, originally just how much can we make and what's the feasible amount and concentration in a solution, and by the limits of a volume that we can give a mouse or an animal, sort of determine our initial dosing, and can we up that and make it higher, and what are we seeing? So the doses were arrived at through animal studies, and sort of doing enough of those to know what's reasonably safe and what is still high enough to give an effect in terms of what we want to see. And a lot of the dosing started originally with intramuscular or localized limb perfusion before we went to full on intravenous systemic dosing. So really it was determined, what I would say, empirically by trial and error and in animal models before going to people. But people are different than animals, and so we need to do some of this dose discovery in humans as well. And so some of these trials, for example, hemophilia, started off with a little bit lower dose and did a little bit of dose escalation within a narrower range, staying within a range that we thought might be effective, sort of going instead of, 1 e to the 13th to 1 e to the 14th, going 2, 4, and 6 e to the 13th, that sort of thing. So, and some of the dosing, I think, in these trials were probably based on the lessons from earlier trials, such as the hemophilia trials. What kind of dosing is being safe in other people for other diseases, and also being effective for those diseases.
Thank you. I actually had an additional question based on something you touched upon earlier, that maybe you could help address, just in case there's any confusion, or if families are kind of hearing about it for the first time. That idea of empty capsids, is that just a byproduct of our current manufacturing process, and in trying to lower the amount of empty capsid being delivered? Is that something that just needs to be improved upon from a manufacturing standpoint, and something we can expect to improve over time, or are there other ways to address that issue?
Yeah, it's interesting. When we make small molecule drugs and we're chemically synthesizing them, we're very much in control of that process. When you're making a virus, although we like to think we're in control, and we do have some control, it's a biologic process. It's a living thing. We're infecting cells with a small amount of the virus, or we're putting plasmids in to cells that create that virus. But the process happens in what is really essentially each small manufacturing plant, one single cell. And each of those cells assembles that virus. It makes the viral genes into proteins that forms the capsid. It packages it replicates the DNA. It packages that DNA into those capsids. And then we have to break open those cells, or take the lysate from the cells, and take that whole gamish, that cells, cell debris, membranes, lipids, other proteins, mixed in with all those viruses, intact virus capsids, both empty and full, and then purify those viruses away from all the other materials. And each time you do that, there might be variations. Biology is full of variations. Certainly, good manufacturing procedures, GMP, and the regulations that encompass those, try to minimize all the variations. Try to have master cell banks that are certified. We do lots of testing to verify the quality of all the agents that go into it, and their purity, and their lack of contamination, and to make sure that nothing else is growing in there at the same time, what we call adventitious viruses that might sneak in. And so that's why we've got these facilities that are super clean and filtered air, and lots of processes before you walk in there. And all the reagents are very, tested and validated, and all that. But variations can sneak in there, that we are unaware of, or that we can't totally control. And any biologic process, there's going to be variability. So the biologic process of packaging of a virus, the normal process, results in these empty capsids. It's just not that efficient. So can we tweak that? We're not in there taking DNA from one side of the room and stuffing it into a virus capsid shell on the other side of the room. It's the cell doing that. So it's really in control. So we can just change parameters and hope and see if those parameters change the ratio of empty to full. We can change the temperature. We can change, concentrations of nutrients in the media that the cells are growing in, et cetera, et cetera. We can change the ratios of our plasmids that we insert. So there's a lot of different things that we can and have been tried in terms of improving on that. And then the purification process, how to separate the viruses from all the other things. In the process of separating the viruses from all the cellular debris, can we separate the empties from the full? And there are certain ways to do that. They have different densities, so you can spin them on what we call a density gradient, and empties will band in a different place than the fulls, and we can take out the fulls. So there are things we can do. So that yes, there are manufacturing procedures that we need to hone and get better at, but it's unlikely to be something that we can do 100% and 100% of the time. And again, some have thought that the empties serve a role in terms of being a decoy for antibodies, and so we have to figure that out as well.
So with all of the variability in biology and, doing all of this engineering to find the right dose and to find the right serotype and to identify all of these parts and pieces, but also keeping in mind some of these adverse events that we've seen in different trials and different diseases, what are some of the things that we can think about to ensure safety as we move forward?
Yeah, first and foremost, the FDA has some great guidelines and rules in place, and they're evolving as well as the field evolves in terms of making sure that uh, people and companies that are taking these products, developing these products, and taking them into patients follow the guidelines and adhere to the best known principles and practices, and um, uh, to ensure safety. I mean, that's the biggest concern of the FDA is safety. Um, Obviously, with any drug, there's a risk-benefit ratio. Almost nothing that you put in your body is going to be completely free of side effects, and we've just lived with that in medicine forever. And there's always going to be a little bit of a trade-off. So the goal is minimize risk, minimize side effects, maximize benefit, maximize efficacy. And so, in our collective humanity's collective strive to get that ratio as good as possible, the processing procedures are going to continue to evolve and change as technologies evolve and change. But one thing we all must be thankful for, I think, is that we do have an agency like the FDA watching over this to make sure that it's done properly. Now, in my more junior days as a faculty member, I often had negative feelings toward the FDA in terms of access of drugs for patients. Certainly in my field, cancer, patients are dying, we need access to the drugs, et cetera. But I have come to be quite thankful for their safeguarding because we don't want to do harm. And they are good people doing all they can to ensure that we minimize the chances of harm. So, but it is important to recognize that the processes and procedures and the best practices are evolving. They're something that we, as a group, together are learning, and they're changing all the time and getting better and better. So the things that we might have been allowed to do in the olden days or to try, we're no longer able to try or do, and that's a good thing because the risk-benefit ratio was not favorable for those things. So I think that's the biggest safeguard. The other thing is, I would say, on the clinician side, in terms of what's the real world experience with patients. And for example, with the mild tubular myopathy experience, we all as a society need to learn what happened. And we need to know what, if the better we understand what exactly happened and why those patients might have been at higher risk for a problem than others, then the more safeguards we can put in place. Perhaps we can discover certain biomarkers or levels of substances in the blood, or levels of function or dysfunction of organs like the liver that are just too much to handle and the risk is too great. So I think the clinical side, observing what's happening with patients and observing and trying to come to a better understanding of what what kind of selection of patients we might be able to implement to make this to minimize the risk.
That was incredibly insightful and I think very helpful. Dr. Kripe, we just want to express our gratitude for taking the time to help answer some of these questions that have been so important and pressing to patients and families in the Duchenne community, and for taking the time to speak with us today. I think that's going to help a lot of families have a better understanding of what's going on in the world of gene therapy. And we look forward to hearing from you again ahead of our live panel this upcoming week in our virtual conference. So thank you again.
You're welcome. It was my pleasure.